Okay, why don't we get started? Welcome everyone to the first instance of the Toronto Vision Seminar this year. Good to have everyone. We've got a big crowd, but we also have an overflow room, so we may even have more people joining. But it's my pleasure to introduce Ellen Zhong. She's an assistant professor at Princeton University, where she has been working on at the intersection of AI and imaging and biology, and she's going to tell us today about some of her work on cryo-electron microscopy. And prior to that, she was a PhD student at MIT, and she also did some time at DeepMind working on AlphaFold. So yeah, really excited to have Ellen here with us today, and excited to hear what she has to talk about. So please join me in welcoming her.
Introduction, and I know a lot of the researchers here at U of T, York, Structura have really developed pioneering methods and software that have completely transformed cryo-EM. And so it's an honor to be here to tell you guys about my work on machine learning for determining protein structure and dynamics from cryo-EM images. So I'll start with the biological motivation, which is that all central biological processes are carried out by proteins and protein complexes. So we want to study these molecules in order to better understand, the fundamental molecules or mysteries of life. We can imagine intervening in medical and therapeutic purposes, as well as engineering new nanotechnologies and biotechnologies. And proteins come in a wide variety of shapes and sizes, and one of the fundamental central tenets of structural biology is that the shape or the structure of these molecules lends itself to its function. And so I always like to highlight the PDB 101 molecule of the month. This month's molecule is the ZOR1 resistome, which is fundamental machinery in plant immune systems to prevent against rot and other kinds of viruses. So fun fact of the day. So since this is Toronto Vision Group, I didn't suppose any biological kind of background. So just for a very quick primer, proteins are biological molecules that consist of a linear chain of amino acids polymerized together. There's about 20 different amino acids. So these sequence representations, there's 20 possible characters. So this is this one-dimensional sequence representation. You might have heard of these like large protein language models. Maybe you could infer a lot of things already just from these databases of protein sequences. But then these one-dimensional chains are actual 3D molecules, and they self-interact to fold into a specific three-dimensional shape. And again, it's the shape that gives the rise to the function of these proteins. And so just highlighting a couple different 3D representations. On the left, this is a full atomic model. So basically, we're kind of visualizing the 3D Cartesian coordinates of every atom in this protein. That might be kind of messy, so there's often a shorthand, which is just the backbone representation with these types of motifs, or ribbon motifs, so that's in the center. And then in the context of cryo-EM, we're often thinking about density volumes, electron scattering potential of the atoms in the system. And so the visualization on the right is this isosurface representation, and maybe given a sufficiently high resolution map, you can fit in the underlying atomic model. That's, 3D structures of proteins 101, and structural biology is the study of these molecules via their three-dimensional structure, and not, their sequence or not other types of biochemistry or biophysics. Okay, so from the computer science perspective, there's been major breakthroughs in silico deep learning-based methods for predicting protein structure. And so we have AlphaFold. So since, I guess, the 2020 performance at Cas14, which is this double-blind community-wide competition, this completely, stunned the community in terms of what was possible from a pure kind of deep learning approach. So that's represented here as this black box statistical machine that's learned this accurate mapping from the input sequence to the 3D coordinates. So this is very interesting on the algorithmic side. And since then, there's been an explosion of, different types of methods for machine learning methods for structural biology. And so I'm just highlighting here ESM-fold, which is hooking up these large language models, protein sequences, to some of the architectures underlying AlphaFold to be able to predict structures at scale. And, there's also the Zetafold, the Megafold, and all this like Starfold kind of suite of methods. And then there's also been very interesting, diffusion generative models of proteins. And so this paper, which was presented a year ago, was just published last week in Nature. And so I just love this paper. I think there's a lot of really interesting methods, and I think it heralds a very exciting new time for kind of generative models over proteins and their 3D kind of data, 3D representations. Okay, But what's next? So how far will these methods go? So I think in terms of the frontiers of machine learning for structural biology, a lot of these methods are ultimately, top-down machine learning methods that are trained on the PDB or the protein data bank. And so I think that gives really nice intuitions in terms of thinking about how far these methods will go, because these top-down methods are ultimately just learning patterns from this database of crystal structures. And so the frontiers I think of are complexes. So these proteins are, molecular machines, they are moving. And so how can we predict or reason about their different conformations, their dynamics? That's been a focus in a lot of our work. Their interactions with small molecules, maybe drugs, and also what is their structure in vivo, in the actual cellular context. So that's kind of depicted here on the right with this, artist's rendition of what's going on inside E. coli. And then one last piece of background before diving into cryo-EM. So some people, they know AlphaFold exists. They're like, okay, that's it. Like, we can understand 3D structure. What else is there? I want to highlight that there's different levels of accuracy of thinking about the 3D structure. So there's this pure in silico prediction on the top right. We also can experimentally solve structures. So this has been the dominant approach, is the experimental study of protein structures for the last many decades, and mostly via a technique called X-ray crystallography, where you crystallize your protein into this into literally crystals, and then shoot it with X-rays to resolve the 3D structure based on the diffraction pattern of those X-rays through the crystal. And then recently, there's been imaging-based approaches via single particle cryo-EM, where we can study proteins in solution. So this is getting slightly closer to what they actually behave and look like. And also very recently, directly inside the cells. There's been really new innovations in terms of the imaging techniques that can image slices out of whole cells, and we can begin to study, the structure of these complexes and what they what their structure is in vivo, or in situ at least. Okay, so cryo-EM has undergone the so-called resolution revolution over the last decade, and that refers to the rapid increase in the quality and speed of this technique. One kind of culmination is the 2017 Nobel Prize in Chemistry went to the original creators of some of the techniques of single particle cryo-EM, and it's been particularly exciting because this resolution revolution has opened up new areas of structural biology. And this has been driven by parallel developments in the hardware and software. So in particular, direct electron detectors that fundamentally increase the amount of signal in the collected images. So I think this was one of the key advances that led to being able to solve high resolution details from cryo-EM. And then concomitant to these improved kind of optics, electron optics and hardware, there's been a suite of new reconstruction algorithms developed by many in this room, accelerated on GPU compute, where the development or the application of cryo-EM kind of imaging processing, which used to take weeks or even months in an entire compute cluster, you can now do in a couple hours on a commodity GPU application. And so at this point, there's been significant automation and democratization of cryo-EM imaging. And I think one thing that's very exciting is that right now we have kind of a lot of new computational challenges and opportunities, since it's now becoming much easier to collect this type of data. So giving a brief overview of the image analysis and image processing pipeline for cryo-EM. So step 0 is experimental step, which I'm completely skipping, which is obtaining a purified sample of your molecule of interest and then imaging it in your very expensive, fancy transmission electron microscope. The raw images you get out are called micrographs, and then there's various steps in pre-processing, so motion correction and particle picking, where you have algorithms that identify each instance of your molecule, draw bounding boxes around them, and extract them as single particle images. Then there's the 2D to 3D reconstruction task. So this is, been the focus of some of our work, which is where you have your data set of collected images, typical number being in the hundreds of thousands, millions, these days even tens of millions. Can we reconstruct the 3D cryo-EM density captured in the data? And then finally, given a sufficiently high resolution density map, or this density volume, we can fit in the underlying atomic model. So maybe digging more into single particle cryo-EM image formation. So this is the Ford model. And again, we have this purified solution. It's flash frozen in a very thin layer of ice and then imaged with our transmission electron microscope. So electron beam is differentially scattered by our molecule relative to the surrounding amorphous ice and then detected on the other end. We model each cryo-EM image as a tomographic projection of this density volume. And so the image formation model is kind of this integral of our volume over the z-axis, where z is kind of the imaging axis. But because the molecules are tumbling randomly in solution before being flash frozen, there's some unknown orientation or this kind of 3D rotation. We also model kind of this in-plane shift, which corresponds to imperfect centering of our molecule within the image, convolution by the microscope's point spread function, or the CTF, and then finally Gaussian additive noise. So that's the forward model. The goal of cryo-EM reconstruction is to solve the first problem. And so there are a number of challenges associated with this computational task. First and foremost, the unknown particle poses. These are tumbling randomly solutions. We don't know the orientation that we're seeing each molecule from within this kind of unlabeled noisy data set. The images are very noisy, so this is actually a pretty kind of high signal data set. Most of the time, you can barely make out any signal from the collected images. You have to take into account these image grading filters, which just makes life challenging, discretization of the measurements, and then the so-called heterogeneity problem, which is where we break the assumption that each image contains identical kind of static 3D structure. Before I get into the heterogeneity problem, just one more piece of background on cryo-EM, which is the Fourier slice theorem. So this tells us how 2D projections are related to the 3D volume. It states that the Fourier transform of this 2D projection is a central slice out of the 3D Fourier transform of the volume, perpendicular to the kind of projection, perpendicular to where the slice is oriented perpendicular to the projection direction. And so this suggests a way of reconstruction. If you have your set of images, if you know their poses, then you can, convert them into the Fourier domain, insert them as slices at their corresponding orientations, and that's for the 3D Fourier representation of our molecule, then compute the inverse FFT and go back to our real space representation of our volume. And so this procedure is kind of how a lot of traditional homogeneous reconstruction algorithms work. It's formulated as optimization of a single objective function to find, maximum likelihood estimation to find the 3D structure or a volume that maximizes the likelihood of the data. And so, given this objective, you can often ascend it with expectation maximization or other kind of iterative approaches, where at one step you estimate all your alignments given a fixed volume, and then at the M step you re-estimate the volume given your fixed alignments. And there are many state-of-the-art software packages that now do this very effectively and efficiently. And so Reliant, which first formulated this Bayesian formulation of reconstruction in cryo-EM, and then of course many of you guys here are familiar with the very kind of innovative methods for stochastic optimization to avoid local minima, and then the whole suite of software that now exists in the CryoSpark for doing 3D reconstruction and beyond. So now returning to the heterogeneity problem, each image here contains a unique molecule, potentially in a different conformational state. So the ability to image heterogeneous structures is actually a major opportunity for cryo-EM. So in crystallography, we're experimentally unifying every copy of our molecule in the exact same structure in our crystalline lattice. But in cryo-EM, we're just capturing the structures flash frozen in their near native states in solution. So we actually get the experimental data needed to study the heterogeneity of these different molecules. Unfortunately, you can imagine how this makes reconstruction much harder. So standard approaches for dealing with heterogeneous data include just ad hoc filtering or getting rid of as much of the heterogeneity as possible, sometimes even returning back to sample preparation. This is obviously not ideal if you're interested in the heterogeneity. And really what the workhorse is for dealing with heterogeneity is something that I like to call multi-class reconstruction, in the field also called 3D classification, which is where you extend the image formation model to assume that images are generated from a discrete mixture model of k independent structures. So that might work well if your protein has a couple of distinct states, but you don't know this number a priori. There might be more complex forms of heterogeneity. And so what happens in a typical cryo-EM processing workflow is something like this, where it becomes this very onerous, expert, user-driven procedure, where at the top you have millions of images kind of picked from micrographs, and maybe with like some upstream filtering. And then through this very onerous user-driven process of 3D classification, where you kind of know just like by expert intuition or maybe just guess and check a lot which kind of subpopulations to take. By the end, you filtered it down to 2, albeit high resolution structures, but you've also thrown away 90% of your images. And so I think I pulled this out of an SI of a cryo-EM paper from 2001. It's definitely still the case that it's this very kind of onerous procedure. And I think I'm very familiar with people who have thousands of jobs in CryoSpark workspaces where they're really just going through and figuring out what's in the data. So the identification analysis of heterogeneity, especially continuous forms, I would say is definitely an open problem in cryo-EM reconstruction. I first learned about this problem of heterogeneity in cryo-EM after reading this paper from Roy Letterman and Singer at Princeton. So they're mathematicians, and I highly recommend you check them out if you want kind of a more formal description of the problem. But our research goals starting out was, can we approach this heterogeneity problem with modern machine learning methods? So can we design this modern machine learning approach for dealing with heterogeneity in cryo-EM? So for the rest of the talk, that was the motivation background. I'll give a quick overview of CryoDragon, which is our approach for heterogeneous reconstruction, and then very quickly talk about 2 new methods in CryoDragon, which is ab initio reconstruction and in situ reconstruction, and then end on kind of some concluding thoughts for ML and cryo-EM, as well as what happens now in the post-AlphaFold world. Okay, so CryoDragon is an acronym, which I'm very proud of. It stands for Deep Reconstructing Generative Networks, and we frame heterogeneous reconstruction as unsupervised learning of a deep generative model of 3D cryo-EM structures that's trained from 2D cryo-EM images, unlabeled 2D cryo-EM images. And so there's a couple of aspects of this method. The first is our architecture for modeling 3D structures using coordinate-based neural networks. The second is the overall kind of machine learning system, which is based on the BAE. And then third is how do we train it with unknown poses and this unknown kind of heterogeneity, unknown latent variable describing heterogeneity. So for the first, one of the key ideas in CryoDragon, especially relative to contemporary approaches, is how we model. So instead of representing our cryo-EM structure or volume as discrete points on a 3D lattice, we instead use a neural network to directly approximate this continuous function, mapping 3D Cartesian coordinates to the cryo-EM density at the location. So CryoDragon structures are parameterized with this neural network model instead of a voxel array. And so the goal of reconstruction in the traditional settings is to estimate the value of the cryo-EM densities at each of these voxels. In CryoDragon, it's to learn the parameters of this neural network. There's an additional architectural detail, which is that instead of directly inputting our 3D Cartesian coordinates, we featurize it using a series of sinusoids of varying frequency. This was originally inspired from the transformer literature. And since this is a vision group, I'm pretty sure you guys are all familiar with a NERF and neural radiance fields. And so something that's been very exciting to me is seeing all of the related work and developments in implicit neural representations and neural fields for very similar types of modeling problems in computer vision. So why did we do this in CryoDragon? The main reason was to model heterogeneity. So in this multi-class reconstruction setting, you would have k separate voxel arrays. And again, there's quite manual selection of k, the initial volumes, which can significantly bias the results of reconstruction. And typically k is fewer than 10, although I think that is now much higher in the latest version of CryoSpark. In CryoDragon, we simply extend the input dimension of our neural network to include an additional latent variable vector. And so this allows us to condition our model on some continuous latent variable describing heterogeneity. So we extend this neural representation with this conditional latent variable model. This allows us to encode this n-dimensional continuous distribution over structures. And so from kind of the structural biology community, if our latent variable were one-dimensional, you can imagine this as parameterizing location along some 1D reaction coordinate. In general, we use a slightly higher dimensional or n-dimensional latent space. You can think of this as describing position in some abstract kind of conformational landscape of our protein complex. So that's the generative model in CryoDragon. The question is, how do we learn this model from our data, which look like that? Right, so unlabeled, very noisy. How do we possibly learn the parameters of this neural model? So our overall architecture in CryoDragon is this Fourier domain image encoder, volume decoder architecture, which is based on. So I'll talk through this briefly. Given an input image, our encoder maps this to latent space or distribution in our latent space. And then given a sample from that distribution or the latent code, which ideally describes the structural state present in that image, and the 3D Cartesian coordinates corresponding to that image. So remember, by the Fourier slice theorem, each image is a planar slice out of our 3D model. We can then reconstruct the value of the cryo density pixel by pixel, render the slice, compare that to the input image, compute the loss, and then use that to update the neural network weights. So the decoder reconstructs an image pixel by pixel given z and the 3D coordinates, and this coordinate-based following architecture very naturally enforces, the geometric consistency between these different 2D views. Okay, that's if we know the coordinates, which we don't know this a priori. And for each image, we need some method to approximate the pose, right? So the pose we explicitly apply as this rotation of a coordinate lattice. But we need to know what this pose is. How do we do this? Where does this come from? So there's a couple possible paradigms that people can answer if they want. There's a couple possible paradigms for pose inference. So maybe coming from the deep learning perspective, the easiest, most naive thing is just train another model, maybe via amortized variational inference. So train another encoder that can predict the pose given the input image. Maybe we can, because our model is fully differentiable, we can directly backprop into the pose variable, so we can initialize it and then directly update it with gradient descent. There's also, been some interesting work on distribution matching-based approaches where we just completely skip over pose inference and try to model the distributional structures and simulate image formation to kind of match the distribution of the data. It's an adversarial approach. Here's a couple of possible paradigms. When approaching some of these with our early experiments, we ran into many kind of spurious local minima in the training objective. And so this is this toy experiment where I take a hand and then I simulate creating image formation with noise and without noise, and then try to train a model to predict the pose, and we run into weird local minima, so that's here on the left. And given some random initialization of the pose, we just definitely don't recover anything meaningful, right? So we ran into lots of problematic local minima. So in the original kind of approach for cryo-dragon, we instead take, a leaf out of the traditional cryo reconstruction literature and perform a global search, or kind of an exhaustive search over a discretization of the 5-dimensional space of poses for each image given the current value of the decoder. And so this has kind of two aspects. The first is this hierarchical search procedure. So we can start with an exhaustive search on a coarse discretization of the space of possible poses, and then iteratively refine the top K poses until we get to a finer, kind of more precise estimate of the pose. You can choose K for the top K poses to keep via a branch and bound procedure, or practice we just use the kind of a fixed value. And then the second technique is frequency marketing. So here we band limit the loss function to low frequency components. And there's two benefits of this. The first is computational efficiency. So because the model is specified in the Fourier domain, if we only want to reconstruct the low frequency components of the image, we only render, a much smaller radius of pixels than the full image. And so it's much, much faster. And then second is to prevent overfitting. So especially at the coarse resolutions of the grid, we're not going to be able to align the high resolution details anyway. And so we want to just avoid rendering those to overfit to spurious noise or have spurious correlations when computing the losses. So with this hierarchical search procedure, then we can recover our simulated protein, which is the hand. So this works, pretty well in this toy setting. But one of the main challenges here is, there's also just many, many hyperparameters in this post-search procedure, and so you can be very fancy in how you engineer that. But one fundamental limitation of the cryo-dragon kind of implicit neural representation, especially relative to traditional approaches, is just the speed of rendering the model. So for any kind of off-voxel grid point in the voxel-based approach, you just take the weighted average of the 8 spatially closest neighbors. But in cryo-dragon, we need to do a full pass-through neural network. So this is many orders of magnitude or flops per point to render. So this is much more expensive to evaluate. In cryo-Dragon 2, which was at ICCB, and this was in collaboration with Anna Blair and my two PhD co-advisors, Joey Davis and Bonnie Berger, we made some changes to the underlying kind of ways we did post-search, push as much of it into image space instead of rendering volumes, and then we can do ab initio heterogeneous reconstruction much faster and much more accurate. So just showing these movies, our original approach, we ran into it took 38 hours, and there's like various alignment artifacts. With ground truth, there are fixed poses. This was 4 hours. That's cryo-Dragon with post supervision, and then in cryo-Dragon 2, we can recover the pre-catalytic spliceosome in less than 12 hours. Okay, so that's great. Recently, we've been trying to iterate on that. So that was still just this preliminary work, and it was still slow, and it was still annoying, and it still had all these hyperparameters. And so we revisited recently this amortized approach, right? Because one of the main advantages here is if you can learn this function, then you can avoid this exhaustive post-search procedure. You can just predict the pose given, just do one forward pass and predict get the pose given an image. And recent methods have extended or have developed have extended amortized inference for ab initio reconstruction in challenging realistic settings. And so Axel Levy and co-authors at Stanford developed cryo-AI, where they posed kind of new loss functions that let you avoid these spurious local minima. And the idea here is that instead of performing a separate independent pose search on each image, we train a model to predict the pose given an image. And this can be trained end-to-end, and this works on experimental data. And so in CryoFire, which was a collaboration with Axel and company presented at NeurIPS, we apply this to the heterogeneous setting and can then do pose search and heterogeneous reconstruction much, much faster than this exhaustive search procedure. Is that good enough? No. So we've been iterating on that more and more. One major challenge in a lot of kind of realistic cryo-EM data sets is that they're extremely noisy, they're very large, and we definitely started reaching limits of this amortized inference procedure. And so what I'm showing on the left is our pose error versus amount of noise, and there's always this memorization gap. And so if we can train the model and apply it on unseen images, then we can really quickly do pose inference. But we always have this, gap, especially for low noise, for high noise, especially for very, very noisy images. The model would tend to just memorize the images in CNET training. And given that our model is memorizing, then it becomes less and less effective for larger and larger data sets. So there's always a capacity gap where, because we have a finite number of parameters in our encoder, we're never going to be able to just memorize every single image we see. So there ended up being also this capacity gap for larger and larger images. And so we definitely start reaching the limits of amortized inference for very, very large, very, very noisy data sets. Okay, so we're turning to this method, like direct kind of optimization, and something that we're working on right now, which is still in preparation, is returning to this gradient descent-based approach. And it's motivated by this question: if our encoder is primarily memorizing images, do we even need an encoder? If we don't care about generalization to unseen data, why use an encoder at all? And so I think one of the research questions is like, how much do we care about memorization versus generalization? Something we noticed that was interesting was regularization becomes pretty important, and is there interesting implicit representation of regularization in this neural model? What we're really interested in is, can we perform end-to-end, robust, scalable ab initio reconstruction on large cryo data sets and get high resolution structures. So very excited about this, and hopefully this will be appearing soon. Okay, so that's ab initio reconstruction. What happens after we train the model? So at test time, we now have these two neural networks. We can use the encoder to evaluate the latent embedding for each image. So we can embed our data set, a million cranium images, into this lower dimension latent space, and then just directly view this data distribution and maybe this something interesting about the distribution of structures in the data. We can also use the decoder neural network to generate volumes given values of our latent variable, and so we can decode different regions of latent space, visualize representative samples. Because we have this generative model with this continuous latent space, maybe given two end states of interest, we can do an interpolation in the latent space of the model and generate structures along the interpolation and get a continuous trajectory of volume space and just visualize the motion of these complexes. And then for validation, so if there's maybe some region of interest, like a structure of interest, or maybe like some strange thing happening, you can always take the images that are embedded into that region, extract them, extract that subset of images, and then perform validation with traditional tools. So like homogeneous reconstruction to see that structure, or in practice, this is a very expressive model, so we model kind of structural heterogeneity, but we also model lots of junk from just false positives that mismatch with the image formation model. So that picked up very nicely, and we can filter the data set. So very useful thing to do in practice. So that's, cryo-dragon, how it works, how we train it, how we're interested in combination reconstruction. Next, I'll give a very quick overview of applications on real data before talking about in situ cryo-dragon. So after developing the original kind of implementation of the method, something that I was particularly interested in is making it actually useful. Like, can we actually reconstruct continuous dynamics of these proteins? I think getting structures is already very hard. There's very few approaches to get the dynamics of proteins. And so if we can actually do that in cryo-EM, I think it's super cool. And so we applied, I think the development of cryo-dragon into a software package really involved, understanding cryo-EM data, which I think a lot of people here can also appreciate. It's just, there's an art to understanding what you're looking at. We apply it to, on the left, is an example of an assembling ribosome where we see what we were able to discover new states that were missed in the original kind of 3D classification-based processing. So that was very cool. And on the right is applied cryo-dragon to this previously published data set of the pre-catalytic spliceosome. And again, the original publication just had a single structure. And so being able to resolve these dynamics and visualize it and compare it to other tools was definitely very kind of fruitful and interesting. So I won't be going to those, but you can check out our Nature Methods paper for more vignettes and kind of a more in-depth story. Yeah.
So in terms of the heterogeneity, it seems to me that there are many different types of changes that can occur, right? It could be articulation, it could be deformation. Is it possible to distinguish between them?
I think not a priori, but one of the advantages of this implicit neural representation is that it's a very expressive model, so we can model all types, even these junk artifacts. And so making sense of it, I think, is another, open problem. How do we interpret the distribution? And how do we characterize distinct compositions, just compositional states versus continuous conformational changes? Yeah. As an aside, I think heterogeneity is already a mouthful, and compositional heterogeneity is a mouthful, and conformational heterogeneity is a mouthful. So I propose calling them compet and compet. So see if that sticks. The software hopefully is relatively easy to use. It's nothing like CryoSpark. It's just a series of command line tools and a Jupyter notebook for interactive analysis. But I wrote a very extensive tutorial on moved it to Gitbook because I saw that CryoSpark was also using Gitbook, and it's way better than the Notion page that I was previously on. And I'm very happy to kind of figure out how to integrate cryo-dragon with other tools like ChimeraX, and I have two research software engineers working with me right now at Princeton on the support and development of this as a tool. I think something that's been particularly rewarding to see is that, or feedback I've gotten is that it's forced a lot of experimentalists like biochemists and structural biologists to learn Python, which I'm very happy to hear, because I think that's a very useful life skill these days in modern molecular biology in 2023. So in the latest version, actually this is slightly out of date because now we're on 3.0 beta, but I won't be talking about the automated conformational landscape analysis, but this is really just encoding some of the best practices that I used when I'm looking at new data sets prospectively, but how do we characterize, different states, and how do we draw kind of continuous reaction coordinates. There's been a lot of advances in computer vision on these implicit neural representations. So we're just taking some of the best techniques from there, bring it back into CryoDragon, and improving the default settings for the positional coding function to lead to faster training and higher capacity models. So in the last bit of this talk, I wanted to talk about some of the recent extensions we've done for CryoDragon to model structures in situ. So there's been very kind of impressive seminal work on applying cryo-electron imaging-based techniques to visualize structures. So this is from Julian Mohammed's group at high resolution, getting atomic resolution structures from ribosomes directly inside the cell. I've also given an overview of CryoDragon, how we can model continuous heterogeneity in single particle cryo data sets. And so some of the problem or the guiding kind of motivation here is, can we apply some of the same techniques from heterogeneity in single particle cryo to the in situ context? So subtomogram, so the way that this works, how you get high resolution structures, is something called subtomogram averaging, which I also want to call it subtomogram reconstruction. It's not really averaging. But the way it works is, instead of collecting a sample of a purified solution, you somehow prepare a thin, like electron transparent lamella, so maybe a cross section out of the cell. So that's shown on the left. And then instead of collecting a single image, you do tomography. So you tilt your imaging stage, collect a series of images, and from that series of images, you can reconstruct a 3D, what's called a tomogram. And then you follow kind of similar protocols, although the tools don't really exist, where you identify all the same instances of your molecule interest, collect that set of single particles, but this time they're called subtomograms, and do high resolution reconstruction by combining all that data, averaging out the noise, and getting a high resolution structure. So instead of a single image here, you can think of this as single particle, but with like a tilt series of images and then a slightly more complicated forward model. So in our recent preprint, which was led by Ramya Rangan, a postdoc in the group, we developed heterogeneous subtomogram reconstruction by extending the CryoDragon approach for in situ cryo-EM data. And so at a very high level, this involves modifying the CryoDragon encoder from just taking a single view to multiple views. And there's many kind of possible architectures that you could do for this. Currently relies on many upstream tools in the cryo-EM data processing pipeline. We also did some software improvements for analyzing large-scale data sets. And I think this is a very kind of, classic story of deep learning, in which some of the architecture design was pretty straightforward, but we really, really had to understand what was going on in the data in order to train the model correctly and get useful results from the model. So this was a very fun project over the last year. One of the interesting research questions, I think, when it comes to heterogeneity in situ is, now we have multiple images. And so instead of encoding a single image, the inference problem in some ways actually becomes easier because we see multiple views of the same conformation. And so I'm just kind of pulling out this early experiments with synthetic data, actually from our original CryoDragon paper, where we looked at encoding 2 images instead of a single image. So assuming you can take a tilt series pair. And then these are just synthetic data sets, but hopefully you can appreciate that the latent space representation of the data set, given 2 images per conformation, is much more qualitatively interpretable than given a single image, right? So we have a lot more information for the conformation that we learn. And so here in cryo-EM, we have a similar setting with multiple tilts. Instead of 2, you practice up to 41, depending on your actual image acquisition. Yeah.
How do you determine the correspondence between this environment and the
That we're assuming is already taken care of. So we know the relative orientations between the different views in tomography, we get the 3D reconstruction, and then giving a single particle like detected from the 3D volume, then we can kind of, I mean, it's very annoying on the metadata to find the exact XY coordinates on each image, but we kind of, from the 3D tomogram, we pick the particle. Okay, so moving to cryo-ET data. This is currently the cryo-ET processing workflow. It is very annoying on the metadata to track all this information, and there's a lot of different tools in different types of operating systems, and it's also very data intensive, and so lots of heads exploding trying to figure out what was going on in cryo-ET. But what was useful was processing this public data set from Julian Mohammed's group, where they were able to identify this 2.5 angstrom structure of the bacterial ribosome. And so we first kind of reprocessed this data set with Positive to Empire, and we're able to reproduce kind of a high resolution structure within the cryo-EM framework. And this is just the standard voxel-based reconstruction. Then we kind of apply the heterogeneity analysis techniques. And so this is what we get. We saw some solar flares here that were maybe these ice artifacts. So that was kind of satisfying. You're like, aha, it's learning something that's not the ribosome. We can easily take out those 50 particles if we wanted to. But we also noticed kind of this region in the latent space that had broken particles right there, kind of messed up. So we can use this latent space representation to filter the data set, remove false positives, either kind of these weird artifacts or maybe something weird that's happening with the underlying data collection and imaging. And then after doing that, training a new model on the filtered data set, then we can recover these 4 different states that's present in the published data set. So they did this kind of, again, onerous 3D classification, and these tools are much less mature in cryo-ET than they are in single particle. And so I think it's like very, I don't know. I've heard this can take months to do. For CryoDragon, this round of training took 2 hours, and I think a lot of, I mean, it pushes a lot of the work into the analysis of the model. But here we can pick out these 4 different tRNA elongation states or tRNA bound states of the ribosome and quantify the abundance of the different states and compare that to 3D classification. We had a second data set in this paper that was a prospective analysis of this large-scale in situ data collected of yeast or S. cerevisiae. I won't kind of go into, and we can, okay, so this was done at 128 resolution. We can take the particles and then do a homogeneous reconstruction of a given state and get high resolution details. This is very satisfying, especially making those figures. It's very fun. So I won't be going to the details of this yeast translational landscape data set, but this was a much larger cryo-ET data set. Here it was 119,000 particles instead of the earlier, which was 18,000. And we were able to visualize the translational landscape of S-cerevisiae ribosomes. So we saw all these different tRNA bound states. We saw these initiation factors, elongation factors. We didn't, I think, do enough justice on the biological side because that's not really our specialty, but this was a very kind of exciting project to just like be able to look at what's going on in situ of inside these yeast cells. Again, we can get the high resolution structures. And I think the really interesting questions from cryo-ET, so right now I'm just talking about the reconstructions of these individual molecules, and we can see kind of the heterogeneity, look at this kind of tRNA channel, visualize the membrane associated states. But I think the really interesting things are relating these different conformations to the actual spatial localization inside the cell. We also have that information. And so I think that's a very interesting biological question of asking how the intermolecular heterogeneity correlates with intramolecular heterogeneity. And some of our ongoing work is really making sense of this like processing pipeline and integrating our approaches for ab initio CryoDragon into cryo-ET data and also validating and refining these states. Okay, so a quick summary and then some next directions. So in summary, cryo-EM, is a very interesting technique. It captures, quote, near-native conformations sampled from capture structures in this near-native state sampled from this equilibrium distribution of conformations, with the quotation marks refer to like caveats for sample preparation. There are now many tools becoming available to study heterogeneity of kind of these cryo-EM samples. Something to keep in mind, and I think makes it challenging but also interesting, is that every cryo-EM data set is different. The choice algorithm encodes different assumptions for the heterogeneity of the sample, and so it's kind of up to the user to understand how these methods work and make sense of how they interpret these methods. CryoDragon in particular has a very kind of expressive model of heterogeneity, and so we've been able to use it to discover new structures, visualize continuous dynamics, filter data sets, but I won't highlight that here. And some of the new and upcoming tools in CryoDragon include our methods for ab initio reconstruction and our in situ reconstruction of subtopograms. So how does this look now in this post-Alphafold era? Since I think there are a lot of people here that work on cryo-EM, I also want to highlight some, I think, interesting areas of machine learning methods for cryo-EM. I think ab initio reconstruction is a very interesting problem. It's very engineering intensive. There's kind of challenges in optimization, maybe room for new methods development, and also, fundamental questions in how identifiable are different conformations when you're also jointly estimating for different poses. The problem of characterizing distributions of protein structure, I think, is pretty interesting. So once you train a model, how do you make sense of it? How do you kind of decide what's comp het versus conf het? Can we develop kind of automated approaches for data analysis? Are there useful benchmarks that we can develop? How do you fit in the atomic models? Like, that's a whole area that I think is like really interesting right now. I think there's a lot more to do in terms of representations and generative modeling paradigms. So we really stuck with these implicit neural representations, but there's all these other sorts of methods that have other types of generative models. And so I think different methods have different types of inductive biases. There are ones maybe like 3D Flex that has better inductive bias for protein motion and dynamics. Can we develop methods that exploit information from sequence or structure databases? Can we encode, okay, this was a workshop paper from a while ago. We're just moving like different, moving Gaussian centers around. And so I think there's a lot more to do here. And then finally, the in situ approach, or the in situ work that I mentioned. I think it's very interesting and amazing that the electron microscope can also be used to capture what's going on, at this subcellular resolution. And can we connect the molecular details to like these larger length scales? I think is very interesting. Okay, so AlphaFold, how does this come into play? The way that I think about it is that right now there's been like 2 parallel revolutions in structural biology. We have methods that can now predict structure, maybe leveraging existing structure databases and sequence databases. I've talked about cryo-EM reconstructions. We have these methods that this is solving an inverse problem from collected images to 3D volumes or 3D structures. And so I think there's a lot to do at this intersection, right? And one method for like structures from experimental data can inform predictions and vice versa. So for example, in structure prediction, it's undetermined, it's an undetermined problem, which conformation you want to predict. So like given the sequence, which conformation should you predict? That was actually relevant in this particular task target for the protein structure prediction community. And so you can imagine experimental data guiding in some way the conformation that you predict. Also, for this reconstruction problem, this is this inverse problem. Can we make reconstruction maybe more sample efficient by leveraging prior information now that we have this like very accurate source of prior information over protein structures? I think there's definitely lots of technical challenges bringing these two together. So one kind of obvious one is the fact that cryo-EM is used to study these gigantic protein complexes that have like hundreds of proteins, thousands of residues, tens of thousands of residues. Whereas structure prediction, like some of these targets are usually a couple hundred residues to thousands. Although I shouldn't be speaking too soon because, like some of these methods, there's been so much progress on these prediction methods. And can they come together to really bring about solving the structures in situ or directly inside cells at atomic resolution. So instead of these artist's renditions of all these different components, can we take cryo-EM data and fill in the details with perhaps like generative models of protein structures and complexes, either trained from cryo data, maybe extending the current approaches, and actually begin to visualize what's going on at atomic resolution from kind of much larger spatial scales. Okay, so with that, thank you all for listening. A lot of the work that I talked about today on cryo-EM and our ab initio has been driven by Ramya and Axel, and I'm very lucky to have fantastic collaborators around the world. So thank you all for listening.
Great. Thanks very much for a really excellent talk. We have some time for questions. Anyone has a question? Okay, yeah.
Really nice one. So there's a second question looking at graphic data about how good spectrum level, how so have you compared the confidential meditation seen from average data versus just the that?
We haven't actually, I actually would love to follow up on that more. One of the original things we found was that you can just run single particle cryo-dragon on cryo-EM data on the untilted version, right, and just reconstruct the distribution of structures. They're low resolution because you only have like a single image instead of like you haven't kind of exposed that many electrons, but you can do that, and that's like quite.
So it's worse resolution?
It's worse resolution just because you don't there's not enough signal in just the untilted, so you still need like a couple of the tilts. But we did do a titration of number of tilts versus just like GSFSC and the homogeneous reconstruction, and we saturate after 8 tilts. And so I think this suggests a lot of room for improvements or difference, like modifications of acquisition for cryo-EM. And like how you spread out your electron dose relative to the different tilts.
So, and then when dealing with volume, of course, each molecule fits itself with different pose and specimen, then has a different, but the high resolution information is preserved in different directions. You would count for that?
In the cryo-EM forward model, we account for it by modeling the dose exposure. And so we just like attenuate the signal the more dose that's seen. So we kind of keep track of like which tilt we're in. But we haven't done anything like super kind of detailed in terms of like anisotropic resolution given kind of different orientation biases.
Is there a question over here? And those of you who ask questions, maybe just speak loudly so that folks in the overflow room can hear. We have this nice microphone array that can pick you up.
I'll try to speak loud. Thank you for the talk, Ellen. Quick question that quick to articulate longer answer. Once you have that liquid space, how do you map that onto some sort of biological development thing, like maybe energy, or which state is the more likely state? Along with that, is there a natural way to kind of define a trajectory, or is that something that's up to the practitioner to?
Yeah, I think this is a great question, and it is not a quick answer. But I think it boils down to the question of how do you define what a state is? And like in some ways, like we have this continuous landscape, right? And so there is no notion of a discrete state. But to make some kind of conclusion in terms of like the probability of being in the state over another, you want to actually like discretize your space in some way and assign like that this one cluster of conformations is a single state. And I think that becomes a question for the practitioner of like what is the relevant, length scale or cluster resolution for the problem, the structure that you have, the protein complex that you have. I see some parallels with like the molecular dynamics community where you run this long time scale trajectory, simulating the physics of the system, and then you want to like discretize that or have like some kind of descriptive coordinate system for the primary modes of motion. But it really comes down to like, how do you want to interpret that ensemble? It's kind of ironic because we learned this complex ensemble, but we want to actually just have k discrete states. So, yeah.
And do you often find that junk stuff goes towards the outer kind of extremities of the living spaces that are common from
Yeah, so that actually is been a trend of if we have like outliers, they're more likely to be they're more likely to be further from the origin. So that is one kind of way we do filtering is just to filter out any like outliers by bias from the origin. But in practice, like I think a lot of feedback I've gotten from users is that they've been able to use CryoDragon to filter out like 50% of their data, which doesn't actually contribute meaningful signal, or that's useful for them because that's like 50%, or that's like half the data set that we need to do their 3D classification on. So yeah.
And they do that by just thresholding that size magnitude.
Sometimes there's interpretable features from the latent space, so you can just directly visualize and then like use that to make your judgment calls. But again, it's very qualitative, and so I think there's more stuff to be done in terms of automating that.
I think if you go back to the slide that shows like the latent space where there was like an isomer fact in
Solar flare.
yeah.
Solar flare.
Yeah. So I guess I was kind of wondering, like, it seems like CryoDragon does a good job here at finding that artifact because radical action is the same defect. Because, on the right, there's like an example of a broken particle.
Yeah.
Were you able to tell if all the particles in that area of space were actually kind of like broken the same way, or are they all could be broken differently? Sort of
Yeah.
Structure.
They all kind of look like this, and we don't know what was going on. In the yeast data set, it was actually more severe, where it looked like all. Well, actually, I don't have it. Sorry. Where everything from this was just this, like there's 25% of the data set that looked like not great, and we want to figure out what's going on. And we thought it was like maybe near the edges of the lamella where the particles where like the sample's been milled, but there wasn't any correlation there. And yeah, I think at some point we're going to give up, but right now we're trying to figure out what's going on.
The colors there, like on this size that resolution, the C resolution. So the first one.
That was just distance from the origin.
Oh.
Solar flare selected, yeah. So this is just this is colored by distance from the origin, and so this is the UMAP. So the origin is like the actual like latent space, latent variable approximation.
Sorry, this is one. The fact that like when training CryoDragon, it's super sensitive to like jump in the data set. Like those things you feel like the particles all the particles like let a certain jump effect of a line in a certain direction in space, but that type of like really contaminate the results you get.
Yes, yeah. So we find training the model is very sensitive to outliers, and one recommendation is just to be very aggressive in removing anything that might be like kind of outlier-y, because I think this has something to do with just like the MSE loss, where like the outliers are like very, very different in terms of the actual like MSE from like the normal consensus structure. And so then a lot of the capacity of the model is spent on these like kind of crazy outliers. Right. And so only after removing that, then we can resolve the underlying heterogeneity. I had some slides on that, but I use those when it's more of a cryo-EM user audience. Yeah.
So in the case of heterogeneity, how do you know you're right? So I understand that you can explain the data, but is it the case that if you look at the same protein, very diverse data sets collected under very different experimental conditions, you end up getting the same sort of data structure, or like how do you know that?
That's another excellent question that doesn't have a good answer, which is we don't know. So there's no ground truth, right? We're trying to solve the structures. We're trying to visualize what the heterogeneity is. And I, again, like to push this off onto the users or the practitioners of like, okay, in the past, the structure was used to like confirm some hypothesis in terms of like how something works. But now, like given heterogeneity, I think it's more used in this like exploratory, maybe kind of generation of hypotheses of like, oh, this might be like how it moves. And then for actually doing some like detailed biological study of a system, you do like all sorts of other experiments to test it. But I think like it's not at all kind of guaranteed, and if you change maybe your like the sample we get is the sample taken at that instant of vitrification. And so if you change the temperature, you're going to change the distribution of conformations. If you change your solvent, you're probably going to change the distribution. If you change it in situ, you're probably going to change the distribution of conformations. Maybe there's some, degeneracy of the actual modes that are accessible for a particular protein complex. That would be nice. That would be very convenient. But maybe it's very context dependent, which I think is kind of one of the driving questions and kind of interesting aspects of structural biology. It's like understanding that context dependence.
Done in query space. And I think your motivation for that was so that since it's a fluid network, you don't have to volume to generate single output volume. And I have to project it out to two different slides.
Yeah.
Which is really great. And then later on, so Andy mentions different other kinds of representations of results. Do you think there are any advantages or disadvantages in terms of the results? In terms of this choice of using query space versus like the simplest thing, fluid space generation that looks more costly, are there significant differences? Did you do experiments to find that there are differences?
I don't think we did any direct experiments on using like an implicit representation on real space data. I think our RBF model might have been in real space just because they're just kind of convenient. It's like easy to do the projection. The major advantage I see of real space is masking, is like the introduction of like kind of spatial priors into your reconstruction. And so everybody always asks for masking in CryoDragon. I'm like, we can't do that. It's like not clear how you do that. In a voxel-based model, you just like multiply your voxel grid with a mask, like binary 1-0 mask. Here, like we could render the whole model, like apply the mask, or like render the model, like regularize in some ways towards like a mask. And it's not exactly clear how one would apply it. So I think that's the major reason why kind of a real space representation is, or a voxel-based representation where you can easily move between real and free. Yeah.
Another follow-up. Also about query space, some of your really amazing results, they show that CryoDragon is able to pull out relatively rare confirmation states in the space. What stops CryoDragon? Space versus a very low population region, different resolutions. Since a query space, when you're doing this, we models actually space. So, but it looks like when you show the videos, all of the volumes across the space have similar like Fourier characteristics and resolution. So is there some explicit regularization that's just that something that just falls out of the model?
I think it's completely like it falls out of the model. So there's just some like implicit like perimeter data sharing between different conformations. So like the C4 state is from this strip particle, 1,000 images. And you just take those 1,000 images and do a reconstruction, you get a much lower resolution volume. But in the CryoDragon reconstruction from this location weight space, it looks kind of high resolution because it's maybe like close to this cluster that has learned the higher resolution structure. And so whether you think of that as a good thing or a bad thing is also kind of in the eyes of the beholder.
Maybe I could just ask one last question. So one thing that is interesting to me is that, well, single particle cryo-EM is just that, a single particle, right? And okay, so we're reconstructing one particle at a time, but do you see a potential for this to grow over like, so that the models can leverage what we learn over multiple particles? I mean, this is kind of what the trajectory that NERF took, right? First, it was just like single scene, basically like photogrammetry for that scene. And then now we have models that are trained on like billions of images, and now you can sample those and recover 3D structures. Like, could cryo-EM follow a similar trajectory?
I think it's definitely interesting research question of like, can we amortize across multiple data sets and like somehow make use of it? Again, I think it depends on the system you pick, and I think there's a lot of tricky parts because we're always applying cryo-EM in a very prospective setting. We're not really trying to interpolate what we know about protein structures. We're trying to really just like solve a new protein structure that hasn't been seen before. Maybe in a drug discovery setting, you're just like doing the same thing and like trying to cover like the same thing, in which case it would be useful perhaps to explore amortization or generalization across data sets. I think one major challenge of cryo-EM is the high noise level. Makes it like that, like traditionally one of the big challenges in cryo-EM is like hallucinating signal or like biasing it towards results because of the high amount of noise in the images. And so I think that's like a major kind of challenge, and whether that can be, I'm not sure whether we can surmount that challenge.
Okay, thanks.
Yeah.
Great. Well, let's thank the speaker again.